morning everyone here. So how are you all? Okay, very good. Now here, this is Radhamal. I have completed my MSc in biotechnology, and here I am going to teach the continuation of your last class. Okay, so let's see. Okay, sexual reproduction, sexual reproduction in flowering plants. Okay, see, previous chapter I have explained about this, right? This one, right? Okay, so I have taken this out. So shall we take this? This is microsporangium. Okay, now I am going to take this part. Okay, this is our target pollen grain from which the pollen is released. Okay, so take this structure out. Okay, taking this structure, okay, so this is rangium, okay, and this is microspore mother cell, right? Microspore mother cell. You have to take this. This is actually a microspore mother cell, MMC. Now take this out. It will be like this. Understand? MMC. This is actually a diploid cell. This undergoes meiosis. That is first meiosis, right? When it undergoes the first meiosis, it the nucleus along with the cell divided into two. Okay. Here the connective tissue will still be presenting. Connective tissue will be present here. Okay. Now again these two cells will go second meiosis. This together is known as a meiosis. Only one meiosis. Okay. So another meiosis happens. Okay. When another meiosis happens, again one cell will form a two nuclei and this one will form another two nuclei. Okay. It will be like this. Understand? Actually this becomes a N. That means haploid cell. Haploid. Okay. Now here still the connective tissue is present. Okay. Still connective tissue is present. After certain development, the connective tissue disappear. Okay. Once the development starts, the developing in the sense is what? It start to develop and the tissue gets disappeared. So 4 separate haploid cells are formed. Okay. 4 haploid cells are formed. This is another one N. This is another N. Okay. These 4 haploid cell is known as a 4 microspore. Just a few minutes. Okay. And each microspore, this is known as a microspore. Okay. Microspore. Each microspore has the ability to form a pollen grain. Okay. Each microspore will develop into a pollen grain to release a pollen. Okay. Now again I am taking this N. okay, which is a microspore. This one undergoes mitosis. If it is undergoes a mitosis, this nucleus is divided into two nuclei. Okay. But the cytokinesis do not happen here. That means the cell will not get separated. It will have the same, it floats in the same cytoplasm. It floats in the same cytoplasm. So it shared the same cytoplasm. It do not divide. The cytoplasm do not divide. Only the nucleus divided. Okay. This process is known as a cenocyte. It is known as a cenocyte. What does it mean? Cenocyte. Okay. Cenocyte. Now, after a further development, one cell will start to form a cells around the nucleus. That means one large cell is able to float the small cell. Okay, so this one becomes a vegetative cell. Understand? And this becomes a generative cell. You know, this vegetative cell do not have any action after this. Okay, but this generative cell has a vast variety of uses. This generative cell will become a future gamete. So here the 60% of gamete formation we have done in the microsporangium, that is male. We have done the 60% of gamete formation, but 40% we are going to see the gamete formation only in the when the interaction happens between a male and the female flower. So suppose just I am giving you an overview. Okay, just see that. Okay, again I will explain you. See, this is actually this becomes a future gamete. When these fall on a female, that is a stigma. When it falls on a stigma, okay, it germinates a tube. Okay, then these generative cells will undergo a mitosis to produce a two nuclei. Okay, this is what here the complete gamete formation will takes place. Okay, so 60% of gamete formation is being taken place here, just outside the cell, outside the embryo, for the microsporangium, for the male. Now we are going to see the formation of female gamete. The structure we have seen. Now we are going to see the formation of gamete in female. Okay, so let me divide this part. Okay. So this is megasporangium. Okay. Now megasporangium is this one, right? Here is our embryo and this is the placenta. This is ovary. This is ovule. This is a style. This is stigma. You know, this is the ovule. Here is the place where the gamete formation happens. So let us take this organ out. Okay, so when you take this organ, it looks like this. Okay, it will be like this. Okay, understand? ovary. Oh, sorry, this is the ovule. Okay, this is the structure of ovule. Now I have to explain the structure first, then we will see the formation of gamete. Okay, now see, this opening is known as micropyle. Okay, then this is an outer integument. Okay, and this one is the inner integument. Okay, and this is a micropyle. Just opposite to the micropyle is chalazal end. Okay, chalazal end. And this whole area, you know, this one whole is known as a nucellus. Okay, so this is the structure of ovule. Okay, here is the place where the gametoformation happens. You all understand? Please listen carefully. Okay, so this is megaspore mother cell. This undergoes meiosis. It undergoes a meiosis. So I am going to do this process outside. Okay, let me do it here itself. Okay. So when this megaspore mother cell undergoes a meiosis, it forms four cells. 1, 2, 3 and 4. Okay, out of 4 cells, the 3 cells gets degenerated. The 3 cells get degenerated, right? So this get degenerated, this get degenerated. Now this is the microspore now, microspore now. It becomes a megaspore. This one undergoes mitosis, first mitosis, and forms 2 nuclei. Okay, it forms 2 nuclei. Then it again go another mitosis to produce 4 nucleus. See, here you can see the cells are not, the nucleus is divided, but the cells are not getting divided. It is floating in the same cytoplasm. The cells do not divided here. Now again this one nucleus, both the nuclei undergoes a mitosis again. So this will produce another 2 nuclei and this one another 2 nuclei. It becomes a 4 nuclei. Okay. Again, it will again undergo another meiosis. Then it will produce a 8 nuclei. Okay, it produces 8 nuclei. 1, 2, 3, 4, 5, 6, 7, 8. Okay, clear. Now I am going to draw this a bit larger. Okay, a bit larger. Okay, please copy this. Copy these steps. Here 3 mitoses had happened. 3 mitoses happened and 1 meiosis. Okay, now please copy this. This is very important. 3 mitosis has happened. Mitosis had happened, okay. So it forms 8 cells, right? Okay, and all the cells are present in the same cytoplasm. These 8 cells becomes a embryo sac. This becomes an embryo sac, okay. Now once the same the nucleus is formed, we are going the nucleus by itself rearranged by itself, okay. Now you see, I told no, it will become an embryo sac. Now this one is the embryo sac, okay. Embryo sac. So 8 cells were there, okay. See here, this one will become our egg. This becomes our egg. And here is another cell, here is another cell. These two will become a synergid cell. Synergid cell, okay. Synergid cell. And this is and 3 cells will get rearranged here opposite to the chalazal end. This is chalazal end, okay. This is micropyle, okay. This is a micropyle end, okay. Through this micropyle only the spore pollen enters, okay. Now how many cells are over? This becomes an antipodal, and here this is, it forms a structure like this. These two is known as a filiform apparatus. The synergid cell will have a filiform apparatus, finger like a projection here. Filiform apparatus, okay. Now how many cells got rearranged? 1, 2, 3, 4, 5, 6 and 2 more here, okay. This is known as a polar cell or a central cell. Now here you see all the cells had formed its cell wall. The cell wall is being generated. Now this one is the most largest cell wall here is formed, okay. Okay, so this is a polar cells, and this one is these two are a filiform apparatus having a projection like a apparatus on a synergid cell, and this becomes a target cell. This is also an egg only, okay. Now the microspore gamete is ready. Formation of female gamete has been taken place. Now our male gamete is ready, female gamete is ready. Now if the female and male gamete is ready, what is the next process? Fertilization. Now see, have you seen this? This is the male, for 60% of the male gamete, right? Male gamete. Now this one, this one, it is from anther, right? So suppose this is the anther. This is the anther, right? This one happens where? Here, this formation, the gamete formation has happened here only, okay. The gamete formation has happened here only. When the pollen is ready, and this is the stigma, right? This is the stigma. When this pollen, the matured pollen, falls on this, sac. This one is stigma, sorry, style, and this is the stigma. Right? This is the stigma and this is style and micropyle. See, now our future gamete is ready to germinate on a stigma. So this is our anther. This is our anther. Now the pollen grain is ready here. Matured pollen is ready. Now it is going to fall on the stigma. Okay. When the matured pollen grain fall on a stigma, what happens? Here the stigma will not simply accept any pollen. It is having some compatibility. Means it will has the ability to select the pollen. Okay. So what happens when the pollen grains fall here? The stigma will has the ability to reject and receive the pollen grain. If it is acceptable, it will accept. If it is reject, it will reject this pollen grain. So how? There is a command between the both stigma and the pollen grain. They will have some interaction. Yes, we should not accept this. Yes, I am your pollen. We have to have the fertilization. This interaction will have between the stigma and the pollen. When this pollen falls, okay, when the pollen grains falls on this, the pollen germinate into tube, pollen tube. So pollen tube, it forms a pollen tube, right? Understand? So it is having already one vegetative cell and one generative cell, right? Understand? Okay. Here, this pollen tube travels through the style by absorbing some nutrient from the style. Okay, it keeps on growing, absorbing some nutrient from the style. Okay, so again it becomes like this. Okay, then it comes like this and like this. Now here two cells are there, right? One is generative and one is vegetative cell. Before reaching to the ovary sac, the vegetative cell gets degenerated. The vegetative cells get degenerated and generative cells undergoes a mitosis to produce two cells, to produce two nuclei cells. Okay. So suppose, see, I don't have, so suppose this is the vegetative and this is generative. This vegetative cell degenerated, gets degenerated, and here the mitosis happens. When mitosis happens, two nucleus cells are formed. Okay, I am very sorry. Please wait. See, I am going to make a micropyle here. Okay, let us change this full structure. See, through the micropyle it is entering. These full structure will be here. This becomes an antipodal, and this becomes a filiform apparatus and synergid cell. Okay, this is what actually. Okay, now you see, once when it enters, no, the generative cells divided into two nuclei with the mitosis. Mitosis happens, okay, in the before entering into the embryo sac. Okay, so when the two gametes are formed, two male gametes are formed, it will enter here. Okay, it will first enter into synergid cell. This is a synergid cell, ma. Okay, once it enter into a synergid cell, it destroy the synergid cell. Okay, and this is the our egg, and one nuclei will get fertilized with this, and the other nuclei will fertilize with this. Okay, this one. Okay, now here you can see. Okay, now this is our, and antipodal will get usually disappear. Now do you all understand till this? Do you all understand? So let us take this part. Okay, so everything is happening here only. I think you all understand this point. Please copy this. This is very, very important. Now antipodal is being synergid cell is being destroyed. Here the two the fusion has happened. This becomes a future embryo, and here this is a triple fusion. This is the triple fusion has happened here. Triple fusion has happened, so it is known as a triple fusion. Okay, now you see these triple fusion will become a endosperm. What it will become? Endosperm. It undergoes this undergoes a lot of mitosis, lot of mitosis to produce primary endosperm nuclei. PE and pen. Okay, it undergoes a lot of so it becomes like this, you know. Okay, this triple fusion was there, no, two polar cell was present at the center that get fused with the pollen grain and forms an endosperm. This is the endosperm, and this endosperm undergoes a lot of the triple fusion that undergo a lot of mitosis to produce a primary endosperm nuclei. And this process here, two fertilization is happening here, one with the egg and two with the polar nuclei. So it is known as a double fertilization, double fertilization. Now you see, this becomes a primary endosperm nuclei pen. This become our future embryo. What will happen? No, these endosperm will produce a food for the embryo to get developed embryo. Now when this start to grow, when embryos start to grow, by utilizing this endosperm, by utilizing endosperm, the development happened. Now you see, first in mitosis, first development happens here in the primary endosperm nuclei, that is endosperm. Because if it is not developing, the embryo is not able to grow, because it has to take the nutrient from here only. Okay, first, so this one is first formed, and then the future embryo will start to use this food and start to grow. Okay, now when it start to grow, first it will form a heart shape structure. Okay, first this is the embryo. It is growing by utilizing the primary endosperm nuclei. Okay, first the development is known as a globular shape, develop into a globular shape. Now again this develop into a heart shape, heart shape. Second stage, this is the first stage. Second stage, you see, this is known as a suspension. The most function of a suspension is to push the embryo towards the endosperm to utilize the food for its development. Okay, next stage, second stage is the heart shape. Third stage, in which the suspension gets disappear, okay, and it start to form horse like a structure. It forms like this. It forms like a suspension. This is a suspension, and usually it disappears. Okay. Now this one will become a future plumule, and this is radical. Okay. So plumule is the plumule will give rise to a shoot, and radical give rise to the root. Okay, so this is how the plant start to grow. Now you see, once this growing embryo utilize all the pen, like a primary endosperm nuclei, when it utilizes, no endosperm is left. Now no embryosperm is left. Then means it has developed into a seed. Suppose the seed, okay, this becomes a seed, right? When when the seed is matured, after utilizing the endosperm, okay, when the seed is matured after utilizing the endosperm, and after even utilizing the endosperm, when the endosperm is left, when the seed is matured, after that also, if the endosperm is left, it is known as albumin. Understand? See, the growing embryo has taken up all the energy, but after taking energy from the endosperm, even if the endosperm is left and the seed is matured, then it is known as the endosperm is known as albuminous. Okay, albuminous. Now this is a nucellus, right? Nucellus. when the ovule is developed into a seed, even after utilizing all energy, and if the nucleus is left, it is known as a perisperm. Perisperm. This is what all about the development, fertilization of a male and female gamete happens. Now next topic will be what? Development of the embryo. This you have to see the next class. Okay dear, thank you so much. Please do subscribe my channel and like my channel. Thank you so much.